Don't.
Good afternoon, sir.
Good afternoon, Dr. Johor.
Dr. Sunil has also joined.
Yeah. Yes. Good afternoon, Dr. Rao. Yesterday I missed both the lectures.
Okay.
Okay.
नहीं मैं तो अब
या
सर वो ज्वाइन करते हैं डॉक्टर साहब का देखा।
नहीं, कल देखना ये था लेकिन वो कुछ काम आ गया, वो देख नहीं पाए कल।
नहीं सर, डोंट वरी, वो पूरा वीडियो शेयर किया है इन्होंने, तो आप देख सकते हैं।
हाँ, अभी देखेंगे। विजेंद्र पाल साहब का है।
ज्यादा स्पायडर तर आपण घेऊन नंतर.
डॉक्टर रोमन ने ज्वाइन किया है क्या?
अभी ज्वाइन किया है।
ओके।
बारिश हुई है क्या डॉक्टर साहब उधर?
हमारे यहाँ तो 2 दिन बस ड्राई हो गया, अभी तक तो बहुत ही अच्छी बारिश हो रही थी।
अरे ये तो बहुत प्रॉब्लम कर रही है इधर महाराष्ट्र में, तो बहुत स्पोरेडिक है अभी।
ज्यादा हो रखा है क्या सर?
हाँ,
बारिश ज्यादा हो रहा है क्या सर वहाँ पे?
बिल्कुल नहीं बारिश,
हो ही नहीं रहा है बारिश ज्यादा।
हमारा डेटा लास्ट 10 ईयर का डेटा है कि हमारा ट्रायल स्विंग होता है।
सर अभी बारिश का बहुत प्रॉब्लम चल रहा है सेंट्रल एंड वेस्टर्न इंडिया में।
बहुत है।
एल नीनो वाला
28 जून तक तो हमारा 80% ट्रायल स्विंग होता है, कंप्लीट होता है। 2-3 जुलाई तक तो 100% होता है, लेकिन अभी एक भी ट्रायल अभी तक स्विंग हो नहीं पा रहा है।
आज हमने तो नासिक में की है सर।
ओके, हाँ, ओके, हाँ, देखते हैं अभी। कुछ स्पोरेडिक है, कुछ जगह पे
हाँ,
है।
कहीं पे
हाँ,
स्कैंटी है रेन इस टाइम जो है।
स्कैंटी है, टोटल स्कैंटी है।
हाँ, वो पूरा वाइड स्प्रेड नहीं है, कहीं पे होता
वाइड
है।
स्प्रेड नहीं है।
तो सर 63 आ गए हैं, शुरू कर दे सर।
हाँ, जैसे भी
आई
एज यू
थिंक
विश।
इन बिटवीन सर वो लोग ज्वाइन कर लेगा, विद इन 3-4 मिनट वो ज्वाइन कर लेगा। अटेंडेंस रेगुलरली 80+ है वैसे तो मनीष शुरू कर दे। शुरू कर। एक मिनट। ठीक। Good evening, everyone. Welcome everyone to day 5 of the training programs. To this program, we'll feature 2 experts as always, and I will be covering the broad theme of understanding the Maize seed systems and how to strengthen it. So it will be largely focused on hybrid seed production, quality assurance, and Indian seed sector overall. And for the first lectures, I welcome our first speaker, Dr. Bijinder Pal, Head Research and Development of Dayal Seed Private Limited. Sir was previously working at Bioseed Research India, where he made significant contributions to hybrid maize breeding. We have heard about many bioseed varieties and their performance, sir. We have already done evaluation and we have conducted so many FLDs of bioseed also. And Dr. Pal is a veteran plant breeder and with over 3 years of decade of experience in hybrid seed development. And today he will deliver an expert talk on seed of genetic potential, enhancing quality assurance in seed production technologies for optimal field performance. So on behalf of all the participants, I warmly invite Dr. Vijender Pal to deliver his lecture. Sir, over to you, sir.
Thank you. Thank you. First of all, let me say thank you to all of you considering my name in such a very important programs which you are conducting. I think this is a very nice way to learn from each other. There was one small things which I like to really tell before I start. I do not know who was the one who selected me for the seed production. I could have been better to give you some better talks in a plant breeding side, but anyway, the seed production is also the very important aspect in the seed systems, and in today era, uh, it's very important to really relook at it our seed system, because my beliefs say that whatever seed systems which we have in India or even sometimes outside, they are all evolved from the self-pollinated crops. And most of the times, the basic principles, the basic fundamentals are very much aligned when you are just simply multiplying the seed of the self-pollinated crops. And in the hybrid, actually, is totally different. So this lecture actually has given me the opportunity to really, put all of my experience together when we are establishing the seed production system. So there are a lot of things which I will be sharing here, which may looks basic, but in practicality, sometimes we don't see them in a field. So this will give you a perspective how the private seed companies basically handle the hybrid seed productions in totality. Okay. Oh, why the slide is not moving? There's some difficulty to move.
slide move नहीं हो रहा.
That's what okay. I have to press it little hard.
नहीं, आप तो आ गया सर, आगे.
So when we talk of the seed productions, actually this is not simply a multiplication. That whatever genetic constitutions you have really incorporated in the F1 hybrid, it has to realize by the farmers. And the seed productions actually has to be placed in such a way so that all of genetic constitutions, whatever you have prepared, it has to reach to the farmer so that it can harvest the genetic potentials of the product, whatever you are producing. And when we are talking the productions, so these are the elements basically you have to keep in mind. There are few which we generally talk, but there are many which sometimes we miss and sometimes we basically, don't consider that much. In today era, the mechanizations is basically, has gone long way, and the requirement of the seed actually has changed. Before it was simply a grading. Simply, using the clippers or sieve to really, separate out the smaller seed and then, some back end noise is coming. Hello. There is some.
I request a colleague to mute. There is some background noise, please. हाँ सर, ठीक है आप ठीक है.
So the breeder actually has to look at it that they are not simply making the hybrid now, they have to think that how they are making a product. And when I talk a product means basically should fit in the production system. It should be produced economically, and also it should be fit to the new mechanization system. These pictures below actually is a real picture from the field where annotated planters has been used, and see the kind of germination, see the kind of the uniformity, and it all can come with the hidden genetic strength of the materials, whatever basically had been bred behind it. So breeding just focusing on the yield is no longer work. It's required to incorporate all of those traits which basically make the hybrid producible and suitable to the new mechanic system. So with this, let me start the next topic, next chapter here. When we talk of the seed, let us try to understand that how many people really get affected with the seed. So botanically, we all know it's a mature ovules, but when it's reached to the farmers, it's not really the mature ovules. This is one farm input which affect the harvest. And all the inputs, whatever he will use, it will be depends on the quality of the seed farmers are planting. So farmers are no longer looking for a seed which he don't trust. He'll make sure that he gets the best quality of the seed so that he can maximize the input usage in his field. Corn has evolved from the livelihood plantings to a cash crop. So each and every plant in his field has a lot of value. So each and every kernel we give him, it has a lot of value, and we have to produce it carefully. So when it go in a production team, it's very different to really look at it because it's a living organism. And this is also the center of the business because we all sell seed. So the genetic purity, seed health, they are all very, very critical to maintain, and they are often get damaged during the production time. Friends, very interesting to see you plant all the commercial hybrid on an optimal condition, on an optimal plant population. The variation will not be more than 4 to 5% in all the commercial hybrid. What's making it different is basically the quality of the seed. If a 95% performance get compounded with a good quality, it can reach to the 100. And if it get compounded with the negative quality, it can reach to the 80. So the production play a crucial role. When it go to the seed processing, which basically in our breeding side, we often ignore because this is not our subject, but this is a very critical subject. Because if you don't process the seed carefully, then it's again damage the germination, and it's create a confusion on a customer side. So we need to be very, very careful on that. When it's go to the quality controls peoples, then they also have to see it's a living organism, and quality matters. If the same hybrid sold by two different companies and they provide a different, the preference of this hybrid will be different, and it will be based on the quality. So in totality, when we say a good seed means it will have a good germination and it will have a better usable plant populations, which will finally value the profit to the farmers, and then it will build the trust on the brand. So when we talk a trust on a brand, actually is start from the quality of the seed. So quality of the seed must be keep in mind, not simply the seed production. So this is basically the beginning of my discussion. So when we talk of this subject, genetic integrity, genetic integrity in F1 hybrid actually is a very critical part, and we often miss it. We generally talk the genetic purity. Genetic integrity, we many times does not really take care it. And when I talk of the genetic integrity, it means the performance of the F1 should never change season after season. I have experienced many times the hybrid introduce something and after 10 years it becomes something else. So genetic integrity is a very, very critical element, what basically has to be taken in mind. And genetic integrity is not really the subject of the seed production team. This is a subject of the breeder. So breeder must understand the genetic purity in a very, very critical way. Maize, as we know, is a highly cross-pollinated crop, and you self it thousands times, it will never become 100% homozygote. There will be always some things there. Yes, in case of double haploids, there are chances. I have not experienced how the line of the double haploids gets sustained over a period of time, or still there are something. But in the conventional breedings, we have seen that maintenance and multiplications, there are two different aspects in the plant breeding. Many people believe in multiplication, but maintenance is a very, very critical element because the residual in a heterotic way, in a heterotic combination, and play a very, very significant role. I have experienced many times the lines is not very uniform, but hybrid is very uniform. And some of my teachers told me, don't disturb the line, maintain as it is. It does not looks very uniform, never mind, don't disturb it, because the residuals is the life of this hybrid. So there are two ways we can follow it up here. When we say maintenance, the continuous selfing actually can kill the line. Continuous selfing is not really a good approach to use in maintaining the inbred line. Alternate selfing, alternate sieving, because it's a population. So you have to maintain the population equilibrium. You have to make sure that whatever you have harvested this time, the next time should looks the same. So this is a very, very critical. And breeder should have a very clear idea whether my line is sensitive for selfing or not. There are many lines I have experienced in my life that are very sensitive for selfing. If you continue selfing them, they start throwing the start throwing the haploidy. They start throwing the barren plant. So I have noticed many times when you do a continuous selfing in inbred line is change. So I'm not sure whether it's written somewhere in a textbook or not, but my experience say that alternate selfing, alternate sieving is the best way to maintain the inbred line, so that you can really sustain the residuals, which play a very key role in the heterosis. The another thing is very important that every cycle you multiply the line, it will shift it little from the original base. No matter whatever you will do it, it will slightly shift in one direction. So this is not really advisable to keep on producing 500 gram seed every season and keep on giving to the next peoples. No. Once the line get commercialized, we should make sure that we have a seed of 5 to 10 kg with us, which can be used for multiplying the parental lines and then go to the next level. If hybrid is a life of 15 years, maximum to maximum, you should multiply the line 5 times. If you will do it every time, the inbred line will get shifted in any directions based on the environment where you are growing the line. remember that this is not 100% how much I got, and the acclimatizations happen. I have a different opinion also nowadays because I start working in a different environment. I see the same line behaving after sometimes totally different in a new environment. And the theory which I was concluding on it is basically there is nothing called segregation. It's basically switch on and off of the alleles which basically required to survive in a particular environment. And a set of the genomic regions get activated to make it survival in a one region, and the set of the genomic regions get set off, which is not required to express in a particular environment. So if inbred lines build up in, say, Dr. Sunil breeding programs and given to Dr. Jahur, and he multiplies every year there only, it will be totally changed after 5 years. It will not be the same anymore. So we have to basically understand this concept, and that is why I believe, and I practice it also, not to multiply the line every year. Once the hybrid get commercialized, multiply enough, and then keep on supplying from the same source. After 4-5 years, just again multiply the seed and then give it from the same source to the others. So that is what basically we do, and most private companies do that. This slide will tell you. Can I remove this ribbon from the toe? Say, okay, yes. Okay. So this slide say how the total seed production system flow. So because in a public sectors, the responsibility of the seed productions belong to the breeders. And that is where basically the private companies and the public sectors basically stand different, because seed production is a chain. It has to go from few to many, and breeder alone cannot do it here. So you have to have a class of the seed. The one which I was telling, don't do it many times, that is class N. So when you have a class N seed with you, you give it to the another function which is called parent seed department in private seed companies. I do not know if the public sector have this type of department or not. So they will multiply the seed which is called O class, and from this O class they will use this O class for 2 to 3 season and multiply the parent seed, and this parent seed go to the production group which they will use for producing the commercial hybrid or commercial seed for sale. So the genetic purity, integrity, the isolation, the qualities are varying in all of this level. There are different SOPs on every level. When you are at N level, it's very different. When you are on level, then it's very different. When you are P level, this is very different. And when you are in a C level or CSP level, then it's totally different. So the one person cannot do all of these exercises. So all of these people's stakeholders actually has to play a critical role in the seed multiplication. Then only you can supply what you have constituted in the F1. So this is just basic fundamentals, because you will be always having a 3 flag in a field. When thousands of the acre get planted, you cannot simply harvest everything and then bag it and then start selling it. No, it will damage the brand. So you have to have a quality control department in between who will give you the flag. When they see the the contamination or the purity standards of the upper block, which are on acceptable level, then they can go ahead and give you the green flag. You can harvest and then start selling their seed. But if the standards are a little lower and they found it that is not meeting the standards, then they will give you the conditional harvesting, that everything, whatever you produce, it has to go through the grow out test. And once the grow out test gets passed, then only you can start selling this seed. And then there is a red flag. If something's found to be very, very old, then this seed cannot be sold to the market at all. So this is a very common practice all the private seed companies basically follow when they produce the seed. It's not that every kernel they produce and they will bag it and they will sell it. And that is where the difference in the performance you will see when you buy the seed of a standard seed companies. So they follow it very, very strictly. There is a lot of efforts involved in it because each and every field of thousands of acre actually has to be put under the eyes of the quality control. So let's one by one. So when we talk of the genetic purity, what does it mean of the genetic purity? I think our senior peoples understand it very well, but the new generation actually has to understand it. When we talk of the genetic purity means the genetics which we have intent to produce should have the maximum in the bag. So any seed which does not belong to a particular hybrid, whether this is from a some another crops or some male lines or some outcrossing, anything which contaminate the genetic constitutions of the F1 basically come in a category of genetic purity. So parental line seed or other hybrid seed or outcross, they all basically contributes in the genetic purity. In most cases, it's just measured by the percentage of the pure seed. And 99% genetic purity is basically the standards. So the quality control inspectors, when they go around, they can assess the field and they can tell whether this is basically the quality seed or not. Very often we have experienced that, the lab test is not really the right way to do it because there are nowadays a lot of talk on the new things, but some things still need to be do in a conventional way, like the MGOT, the people talk MGOT, the molecular genetics, molecular marker growth test. So molecular marker growth test is basically the background is specific, a combinations of the few. It can detect only male and female, but it cannot really detect the whole way of contamination. So the field test is always better. So GOT is always the best. In most of the crop, basically 0 to 0.1% contaminations are allowed. Or to really have a good quality seed, people generally believe that 99% genetic purity should be there. When we talk of the matters of the genetic purity actually in hybrid seed productions most of the times get affected by the isolation because there are another field planted side by. So border row actually play a very significant role to really minimize the contamination from the neighbor plot, whether it's a hybrid seed productions or commercial seed plot, commercial crops. So 200 meters generally consider that one border row is good enough to really manage the little outcrossing from the backside. But sometimes in a field, if this distance are getting narrowed down, then you can manage it by increasing the number of border row. So this is a scientific study done by some of these peoples in Indonesia and Thailand. They did this exercise and then found that if the isolation distance is just only 187 centimeter, 187 meter, then two border row can help. At 175, you need to have 3, and 160, you need to 4, and 150, you can manage with 5. But after that, you cannot control. So this is one thing which is because all the seed productions are in few designated area, and everybody is there. So sometimes it's very difficult to get the right isolation. So to manage those shortcomings, you can increase the number of border row. This is the another topic which is basically very interesting to know because it has progressed a lot. So when we talk of the nicking, in the hybrid seed productions, nicking is also a very, very critical element. So nicking is basically, staggering the plantings. When you stagger the planting, you are making sure that your male and female should flower on the same day. So how to communicate this nicking? I do not know how you people do it, but generally in a private sectors, we start with the 0 and first is the female. So 0 means female day of planting. When I say 0 minus 3, it means the male is planted 3 days before the female. If I say plus 3, the male is planted 3 days later than the female plant. And if 0, 0 is the same. So general principles of the nicking basically is very simple, and if you are not following, please try to do that. There should not be any silk emerge in a field before the pollen setting start. That's the basic fundamental. That no silk should be appear in a field before the pollen setting start. Because if pollen setting if there is a silk emergence before the pollen setting, the hungry silk can receive the pollen from a far distance, sometimes from a kilometers. So it's very critical that male should start first before the silk emergence in a field. Nowadays, mechanizations are happening and farmers are not willing to plant male and female on 2 different dates. They want to plant on the same day. Farmers are start refusing if you are saying that my nicking is 2 days. He says, sorry, I cannot plant it because how I will go for second day again in a field to do the planting. I have just irrigated my female row and you are asking me to go back again second day and do the one more planting. No, I will not. So generally they don't accept anything which does not get flowered on the same day. So the breeder has to make sure now that those days has gone when male and females were flowering on a different days, and you are managing the nicking. Now, commercial farmer, they want to have a male and females are flowering on the same day. That is very, very critical. And US and all, way back, this theory was already implemented because they were planting male and female with the same planter machine. So they don't go for planting again and again. They were making sure that male and female flower first. Plus, they also observed one more, they also innovated one more, element, which is they call it pollinator axle. So pollinator axle is basically it can delay the germination. You treat the seed with the pollinator axle, it will delay 4 to 5 days the germination, and seed will remain healthy in a soil. So when you have your male are early and you want to plant it late, then better to treat it with the pollinator axle, and then you can plant on the same day. It will just germinate 5 days later. So this exercise are very nicely used in USA. There is one company, Infotech in Aurangabad. They are trying to do some experiment for the same this chemical in India also. Some companies are doing it already. So if any one of you are interested to see the effect, you can also try to reach them. They can provide some samples for that. So nicking is going to be the subject which nobody will discuss after a few more years in a cone, because farmer will not take anything where male and females are not flowering on the same day, or male or female cannot be planted on the same day. So this is very, very critical. And this is also very important because when you are planting male and female in 2 different days, the crop management is also differ. The sometime there is a difference of the 5 days in a crop maintenance, so you cannot go back and then do the fertilizations in a female row different day and then male row different day. So this is very important to really have a very little difference in a flowering of the female and male. So there are 6 steps in the seed production. So let me put all of them here one by one. The first step is basically to understand the nicking and also to understand the power of the male and female. So very often we heard that 4:1 is a good male female ratio, but this is not really the right all the times. You can have a male which can pollinate 6 female also, even if sometimes you can go up to 8:1. The important things which basically always get mixed or always get missed when we do the breedings. Our main focus on the F1 performance. My F1 should give more yield than the competitor or than the Czech variety so that it will get notified. So our objective is always on the F1. I think in a commercial breeding, this should not be completely on F1 performance. It has to have a bundle. My female has to be a very high yielding female. My male has to have a very huge pollen ability so that I can plant 6 row for one male, or I can plant 8 row for one male, so that my seed cost will really get reduced. So this is very critical. There are male which can easily produce 6:1. So this is very important. And isolation distance, I told you already there. Sometime time isolation is also work, that is 21 days. The step 2 is planting the male and female row on the base of the nicking study, which is very critical. Here, most of the time we depends on the informations given the breeders, which is not enough. There is a department here which has to intervene. The department name is called Seed Production Research. So once the hybrid gets commercialized, the seed production research guy will go in a field in the area where the material will get planted, and he will make sure that the nicking get adjusted based on the flowering times, based on the reactions of the male and female in this production environment. Because the inbred line produced in some area, when they go for a production in a different area, their behaviors will be a little different. So it's very important to study the nicking before we commercialize the production. Rogging is also very critical element in any volunteer plant or any off-type plant. Unwanted plant need to be removed before the flowering start, not after the flowering. Many times we have seen it, oh, I removed it, but we remove it after pollination. When the pollination happens, the damage is already done. So they're removing the off-type plant actually has no meaning. The another subject which is very important is the detasseling. So detasseling, removing the male flower from the female row is basically the detasseling. It has to be done before the silk emergence. Otherwise, you will have a self plant. But how you will determine it? When it will be done? Is a very critical. Many times we talk day. I think we all scientists in India has to really, somehow work on this concept. Days are not the right indication for flowering or maturity of the hybrid. I think the growth degree day are the more consistent because days will vary. The line will flower in 55 days in one place. It may flower on 75 days on another place. I do not know if someone of you has experience, but I have experience. When you are shifting the male and female from one location to the another location, the time required for their flowerings are differ. They are not reacting the same way. This does not really means that 55 days of the male will be 55 days of the male in the another area. It will be totally different. The second
Yes,
one,
we do experience this when you take it
I'm
from
coming
Srinagar to
here. Yeah, this is the one to go. So the best things to tell the stages. And my experience say that V12 should be considered as a best stage to do the detasseling. Actually, I do not know if someone had discovered it also, but this was basically my research when I see the V12 is the best stage to start the detasseling. And the reason is the tissues of the flag leaves are little tender at that time and they're very easy to remove. When you start the detasseling on 13th or 14th leaf, the tissues are getting harder and the labor has a difficulty to really pull it out. So this is very, very critical to really, try to standardize. If the detasseling start at V12 stage, this is as one of the easiest detasseling and very, very effective because tissues are tender and silk will emerge after 7 days from here. So this is one thing which is very critical to be really considered. The another things which is also very important is the male chopping. Male chopping soon after 12, 15 to 20 days after detasseling, it has to be removed from the field. Sometimes we consider that farmer will harvest something, but that is really not a very good idea because it will mixing of the male will happen, and germ plant security is also one of the considerations. So I think this is very critical to really follow the male chopping after 15 to 20 days of detasseling. And then finally, harvesting. Harvesting is also very, very critical. We should be very careful harvest moisture at harvesting. Anything you will harvest more than 30% moisture, you will be having a problem. Also obtained the certificate from the QC that whether it's a green flag or it's a yellow flag or it's a red flag, so that accordingly you can basically bring the materials to the dryer. How much time I have been allotted? So I will speed up accordingly. So this is, yes, please.
Sir, 45 minutes, sir, but you can carry on, sir.
Okay, so I will try to finish it in 45 minutes. I done already. Oh, sorry. There's only 3 minutes left. So let me see. So this is one of my hybrid. You can see how many rows are there. This is 6:1. Male is too powerful. It can produce 6:1. So I will not go with this one because anyway, this is a basic subject. You all know it. This one I will put a little point here because this is very critical. Seed drying and moisture is basically very, very critical. So when we do a mechanical drying, this is very, very important to maintain the moisture below 42 degrees centigrade in a dryer. If the moisture is above 42 degrees centigrade in a dryer, then your seed will be damaged. Your germination will go down. This work when your moisture at harvesting is not more than 30%. If your moisture at harvesting is more than 30%, then first 2 days you have to dry the seed a little more gently and you have to maintain the temperatures of the dryer at 40 degrees centigrade. So finally, you have to bring down them up to 10 to 12 degrees before you start packing the seed. This one also I can escape. This is very important. I like to really give some few minutes here. When we talk of the germination, it's very simple that every things whatever you plant and it gets germinate is germination. But many times we misunderstand the viability and germination. So viability is basically the seed is alive. But vigor is something which basically make the seed to germinate. Same percentage of the viability or the germination in a very cohesive climate does not create the similar plant populations in a field. Vigors matter, and this is very critical. I do not know how many of you have experienced vigor is a genetic trait, and breeder must be carefully selecting those materials which has high vigor. Many times when you go in your F3 population, you will find out so many plants so many row are not germinating. Few plants are germinating, one or two. And when you start selling them and making inbred line from out of them, they sometimes give a good hybrid, but you will fail in the usable plant populations. They will never work. So this is very important. So when we talk of the vigor, it all depends on the element in a field. So in what kind of environment you have produced, what type of nutrition you have given to the female rows, what is the moisture at the time of harvesting, how is the seed size, mechanical damage, harvesting and processing, and on top of it, the fungus at the storage, duration in storage, and genetics. So these are the elements one has to really keep in mind when we talk of the germination. So germination is not equal to the viability of the seed. Germination basically is viability plus vigor that will make the germination. Seed moisture is a very critical element, and it really damages the health of the seed. So whether it's at the time of storage or whether it's at the time of harvesting, the moisture play a very, very big role. So 10 to 12% moisture at the storage is good, but it has to, if you are storing it, it has to be stored in a low temperature and dry. The formula is 50-50, 50 degree Fahrenheit and 50% relative humidity is the best conditions for the storage. The health of the seed is basically depends on completely the moisture which is at the time of harvesting, as I said it before, or at the time of processing, or at the time of packing. Anything you are packing about 12, life will not be more than 3 to 4 months. So we have to make sure that our seed get dry properly. This is not only for the processing the seed, but this is equally important for our building material too, that we have to really, dry the material properly and store it in a dry places. So quality of the seed, basically, there are two things which has to be really, considered before planting and after planting. So before planting, moisture at harvesting, drying system, cellar you are using, the seed treatment you are using, the packing moisture, and then storage conditions. They are the one who will basically influence the genetic seed quality. But after planting, these are also the factors which will affect the germination, the soil temperature, soil moisture, seed depth, chemical injury, or conditions of the seed planting, insect damage, or any other type of the soil parameters which can basically affect the germination. This is what basically we generally pack all private companies. The germination should be 95% plus, moisture at 10, physical purity 98, and genetic purity 99. So I think this one, I can just leave it because this is not really very critical. This one I like to share. This is my private slide. Don't copy it. When a breeder's giving the material for the production, he has to provide the production technology. In a production technology, he has to give a very, very detailed information to the production team so that they can make sure that every element they consider. So plant height, ear height, silk color, all of those things, if you can add it with the pictorial, nothing like that. So these format actually has to go to the production team when you are sending the inbred line for the production so that they will not get surprised. So I think with this, thank you very much giving me this opportunity to work with to really share some of my idea in the seed productions. I hope it will be usable to all of you. Now, if there is any questions, I will be happy to really answer it. I think I can stop sharing. We can see the screen now. Oh, sorry.
Thank you so much, sir. Questions?
How you will go stop sharing?
Mr. Birendra is saying, sir, this is one of the best presentation that he's having in this 21 days training program, sir.
Oh, no, don't tell me that. Thank you for the appreciation.
Yeah, sir, wonderful as usual. There are a lot of young colleagues we used to go to Hyderabad to meet sir just to discuss maize. I have gone many times with no other work to go to his residence. So please take benefit of his enormous wisdom. Please, who are in the group, our youngsters, ask whatever you or you can write also in the group, please.
So you can personally you can ask also.
Yes.
Aditya was posting some questions. Aditya, you can ask directly to the sir.
Some
Okay,
answers
good
I
evening,
gave,
sir.
but I will like
Good
Aditya to
evening.
ask the same to sir also. Sir, he is asking about modified single cross hybrids, A into A dash into B. How to
Sir, let me put the question. Thank you.
Yeah.
Sir, actually, I wanted to know from your experience. Let's say if a company, if a private sector company is trying to, have a modified single cross hybrid in the market where one parent is a sister line cross. In that case, how do you maintain those lines apart from selfing? The ideal way is to keep selfing them and make the sister line combination and then give it as a parent to the production department. My question is, apart from pure selfing, do you have any other practical method which you have followed to maintain such lines?
Very interesting questions, and I have experienced it already once, because one of my hybrid, it was never been noticed that is a modified single, that was 9909 in Philippines. So actually, sometimes we have to be very clear in the definitions of the modified single cross. When I talk about modified single cross, the lines should be originated from the same F4 plant. That's the precondition. Otherwise, you will not attain the uniformity as you can see it in a single cross hybrid. So in a modified single cross hybrid, if the lines are separating from the F4 plant, then only you can really name them as a sister line. From the F3, the variation is too huge, the line will fall apart. F5 actually the best, but from the F4 itself you can do it. Now coming back to maintaining those lines separately, because they are the sister lines, and if you will continue selfing them, they will really go closer and closer. So the important part here is that you have to understand from the first stand, when the lines are reaching on the F6 level, whether they are further showing the inbreeding diffraction. If answer is yes, then you have to be careful because the line is sensitive for selfing. And hybrid made out of it, it will be totally different the one which you have selected. So in this case, the best things to do it, one season selfing, another season sieving. One season selfing, another season sieving. So you are maintaining the population equilibrium. That is what basically I used to do it. Most of the time what happened, because when we have a bigger programs, we generally, plant some multiplication in a side and ask the labor to do the pollination. And most of the time the selfing looks more easier to them. So it happened to me once. There was one line sensitive for selfing, and when I go to the commercial field, I see a lot of barren plant. They say, oh, from where this barren plant has come? Then we realized that the line was shelved by mistake 3-4 seasons, and then it has started throwing the barren plant. Then we again reconstituted by sieve mating, and then the barren plant problems was resolved. So I hope I have answered. So alternate selfing, alternate sieving is basically the best way to maintain the integrity of the line.
Thank you, sir. But how do you, recognize that these two are different groups and we have not lost the heterotic or combining ability part yet?
That is what I'm saying. If they are separating from the F5 or F4,
Sure.
so they are basically originating from a same breeding cross.
True.
Which already been confirmed the rhetoric pattern in your previous top cross, which was made on F3.
Got it, sir. Now it is clear. I had another question in which I asked, is early vigor, let's say at 15 to 20 days after sowing, a true indication of hybrid potential? Because it might be truly basically just because the hybrid is early. So it will definitely show early vigor compared to late maturing group. So if the genotypes are not segregated, if the hybrids are not segregated based on the maturity group, in that case, how do you interpret early vigor? That is my question.
Actually, there are two things here we have to really see it, and I think you are very correct, because the growth pattern in a plant is always based on the maturity requirement. So if the early maturing hybrid, they generally grow a little faster, but the late maturity groups also go a little slow. But my friend, you should understand also, the late maturity groups does not mean that they start speeding up later, because they have to accumulate more biomass. So generally, if you will go and read the seedling vigor on 20 days, that actually you are not reading the seedling vigor, you are reading the maturity difference. The right days of recording the seedling vigor is before 15 days, before fertilizer application. Earlier, you will go in a field, that is where you can find out the germinability and the vigor of the seedling. We generally go in a field after 20 days after planting. So if you will go 15 days before, you will find out a very, very clear difference between the vigor, and that vigor does not relate to the maturity, because that time the plant has not start moving towards accumulating the biomass.
Now it is very clear, sir. Thank you so much. I have one quick question, totally unrelated to today's topic of your discussion, but I'll make it quick. Sir, in general, for let's say line improvement or line recycling, improvement of existing good parents, in established, well-differentiated company, you have a separate pre-breeding department. So they do that part, trait integration part, and you get the upgraded version of the same line. But as an individual, if you want to make it quick, as an individual breeder, if you don't have a long-term running program where you are going for recurrent selection and you are continuously churning out your inbreds, in that case, what would be your suggestion for trait integration in an elite line?
This is actually, I don't know whether I can really explain you in a 2 minute. This is a very, very, very big subject. And in my understanding, I think it's very difficult to say that this practice is right or that practice is wrong. The point which is very important here is that when you are starting a program, your approach should be different. And when you have already established program, then your approach should be very different. So when you have already established programs, your rhetoric patterns are already set. So most of the times recycling of the lines are become very, very easy. And then sometimes when you start recycling those lines, then you are start finding some another rhetoric patterns also. But in a programs which is basically at very early stage, so the knowledge of the materials which you are using at an early stage is so critical for recycling. So suppose if you are using the materials originated from the same region are the best material to recycle. because they give you the faster outcome. If you will start recycling any one of your old materials with a very, very, different taxas, then your recovery will be very, very low. So my experience say that when you are, suppose I'm in a Philippines and I want to use some material from the Thailand, so I will not use it for recycling that materials immediately, because that will damage the local adaptations of my material. So I will try to make a combination between Philippines by Philippines and Thailand by Thailand, and then only develop the line.
Thank you, sir. Thank you so much. Thank you, everyone.
Next time we can discuss more.
Any other any other questions? Quick, and we have to start the second session. Also, you can have some one question. We can take one more questions. Zahul sir, any remarks, sir?
अरे nothing, he is my guru, no remarks.
Yes, sir.
अरे यार वो गुरु है हमारा.
If there is no further questions, sir, I thank you so much, sir, for an excellent presentation, sir. Your insightful explanation and practical example were very insightful and very engaging, sir. And with the likes and with the emojis that we have seen from the slides and from the from this, we can say that we can say that it was one of the best presentations we ever have in this 21 days training program. Thank you so much, sir.
Thank you so much. Thank you. My pleasure. Okay, so I can quit now.
Yes, sir. Thank you so much, sir. Once again. Chandra Mohan, sir, is there over? He's in. Yes, sir. Yes. So our next, to start with, we have taken some more time, so we have to start quick. So our next speaker is Dr. Chandra Mohan Chauhan. He's a senior scientist at ICAR-IIMR, and he's currently posted at our Resonance Center, Begusararai. And sir was serving at ministry at ministry, and he recently joined at IIMR, and he is basically a seed science and technology. His expertise is in seed science and technology, and he will be covering the topic overview of Indian seed sectors and hybrid seed production in maize. So on behalf of all the participants, sir, I request you to deliver the lecture. Over to you, sir.
Thank you, sir. Thank you very much. Is my slides are visible?
Slide
Chauhan, kindly put it on slide share, and your voice is also not that clear.
Okay. I can use the voice first.
Slide mode and sir voice एक बार check कर लेना.
Voice अब ठीक आ रही है?
अभी थोड़ा ठीक आया सर. It can be increased, sir, I think.
Okay. Maximum है सर. अब देखिए, अब ठीक है?
No, Dr. Saab's voice is not that clear.
एक मिनट में इसको हटाता हूँ उसको ना, फिर बताइएगा. अब बताइए, अब ठीक आ रही है?
हाँ, अब ठीक है. इसको आप सिर्फ स्लाइड मोड में कर लीजिए.
हाँ, वो स्लाइड मोड पे सब मैं कर देता हूँ.
हाँ, प्लीज.
So thank you, Dr. Saab. Thank you very much. And our previous speaker has given an overview of the hybrid seed production in detail, and he's very experienced one. And I had very little experience in case of hybrid seed production, but I will give you an overview of Indian seed sector and how the government sector works in the country. So basically, I would like to start my presentation with very quickly, so that the time is less. So as you know, the seed is very important material for which we all are working to maintain the seed, and seed is basically a reproductive part of the flowering plant capable of developing another plant. So जो seed है उसी के around सारा हमारा जो seed sector it revolves around the seed. So basically the capacity of the seed to reproduce the plant in the next generation is the main thing which everybody is concerned, so that the farmer should got the good yield and productivity. The key components of the seed, we all know that the embryo, seed coat, and endosperm, and embryo is the future plant which helps in generating the root and shoot and other two seedlings. Seed coat protect the seed from dehydration and other things. Endosperms provide the nutrient to the seed. But seeds is vital for the reproduction, dispersal, and survival, and can remain dormant until the condition of preferred. There are two types of seed: monocot and dicot. This is a basic overview of the seed, and the importance of the seed we know from the very beginning. And in our ring Ved also, it is written that Subijam Suchetra Jayate Sampadati, means if a farmer is going to get a good seed, that he will going to get a good yield, and the prosperity will come because of the good yield. He will going to get a good productivity, and more income will be there. So if the farmer used to going to get a good quality seed, the productivity automatically increase from 50 to 20%. So this is the overview of the what is the basics of the quality seed, and what is the importance of seed in the system. So as we speak about the government seed system, so basically there are 4 classes of seed in the government seed system. Sir has discussed about the private seed sector, but in the government seed sector there are basically 4 classes of seeds, and the nucleus seed, the breeder seed, the foundation seed, and certified seed. In all the crops in which we are taking up the seed production under the purview of the seed certification agency, basically there are 4 types of seeds. The production of nucleus seed and breeder seed comes under the purview of the concerned institute and the breeder who has developed the variety. So the breeder, the nucleus seed is developed. The whosoever the breeder is there, he has the responsibility to provide the nucleus seed to produce the nucleus seed as per the requirement. And similarly, after the nucleus seed, the breeder seed is produced under the supervision of the breeder and the institute who is responsible for the development of the variety or the hybrid. The foundation seed and the certified seed is being given to the different agencies that are responsible for the seed production of that particular crop in the country. So the foundation seed is being taken up by the different types of agencies the breeder seed is being taken up by the different types of agencies in the country, like the National Seed Corporation, the State Agriculture Department, the State Seed Corporation, other central seed producing agencies for the reproduction from breeder to foundation and foundation to certified. And all this foundation and certified seed is being produced under as per the standard prescribed in the Indian Minimum Seed Certification Standards. So these are the 2 certified seeds, the foundation and certified seed, that is being produced by the different types of agencies, and ultimately the certified seed is being provided to the farmers. So this is how the government sector works. So another class that the private agency used to multiply is the truthful label seed. The truthful label seed generally is basically of 2 types. The truthful label seed that is being produced from the produce of the notified varieties without the supervision of the seed certification agency, and the truthful label seed produced by the research of the research varieties, which is generally done by the private companies. So private companies are basically, in most of the cases, is doing the multiplication of the research varieties in the name of the truthful seed. But all the seed that is being provided to the farmer should meet the seed standard prescribed in the Indian Minimum Seed Certification Standards. That is a standard developed by the Government of India. So the seed system, basically the entire seed system of our country basically divides into different parts. First, the creation of variation of variety development. First and the foremost part is the development of the variety. The variety is being developed by the different methods, including the domestication, introduction, germplasm collection. In different crops, the different methods are adopted, and the varieties are developed. Hybridization, mutation, polyploidy, monoclonal variation, genetic engineering. So creation of variability and development of the variety is the first and the foremost task in the system. After that, the selection takes place, evaluation, release, and notification by the Government of India. The notification and the release process is under the Government of India, and the multiplication distribution. This is the overview of the seed sector, how the development of the different varieties takes place, and how the varieties ultimately reaches the farmers. So the first part that is there is being done by the research institutes like SAUs, CAUs, ICAR Institute, private companies, and the multiplication and distribution work is under the purview of the different seed producing agencies. In case of private agencies, this both part is being taken up by the concerned private companies. But in the government sector, these two parts are divided since the systematic seed production of the different crops started early in 1960s. These two part of development and release and notification of varieties being dealt by the different types of institutes that are basically the research and development institutes, and the multiplication and distribution work of the seed is being done by the different types of agencies that we used to call the seed multiplication agencies. And in India, there are different types of seed multiplication of agencies are working, which I'm going to discuss in my upcoming slides. The seed system in India, release and notification. So there are different types of person or the institutes or the companies that can develop the varieties. The national institutes, the farmers, SAUs, individual plant breeder, private seed companies, they can all develop the varieties. And after the varieties can be developed, they are put it up in the annual workshop and identified. And after the identification, they are subjected to 3 years of trials. That is IET, AVT first, AVT second. After the the varieties identified the committee, if the variety is found superior to the existing varieties in terms of the production, in terms of the quality, in terms of the disease resistance, if the variety that is being developed by the different institutes or the agencies or the private companies is found superior to the existing varieties in 3 parameters, either it is production, the production, the quality aspect, or the disease or insect resistance, then it is the proposal is for the notification is
kindly mute yourself or accept the speaker, please.
So it is being put up to the central subcommittee. There are 2 subcommittees, one that is dealing with the agriculture crops and one that is dealing with the horticulture crops, and that is being formed by the seed division of Government of India, Ministry of Agriculture. So the different proposals of the release and notification comes to the ministry, and when the committee find it suitable that the varieties are suitable for release and notification, they agree for the notification and the varieties are notified. And then only once the variety is notified, then only the certified seed production can take place as per the Indian minimum seed certification standards. So notified varieties can come into the seed chain. And in case of private companies, they are generally refrain from, in most of the cases, they generally refrain from getting the notification, because they believe that the customer is the king. When the customer is satisfied with the variety, they generally directly go with the research varieties and produce the TL seed. But even if the TL seed is coming in the market, it should meet the standards prescribed by the Government of India, that is the seed standards. It should have the prescribed germination percentage and prescribed purity on the label. It should be tested by the recognized lab, and it should meet the seed standards before coming to the market. Because our Seed Act says that the certification is voluntary, but the labeling is compulsory. Our certification, if you want to get the seed certified, it's on your will, but the labeling is compulsory. So when the variety, whatever the kind of seed that you are selling in the market, it should be labeled, and the labeling should be according to the standards developed by the Government of India, that are the seed standards. So the government seed sector works. So when the variety is notified, and there is a requirement of the seed of that particular variety. So the different the types of agencies that are doing the seed production comes to the seed division, that is the nodal point, focal point in the country, that takes up the indent of that particular different crops and varieties 5 times in a year, and compile the indents of different crops from the agriculture department, from the seed associations, from the cooperatives, national level agencies, FPOs, whosoever is working in the field of seed production. They comes to the Government of India for the requirement of breeder seed of that particular variety, which is recently notified, or even the earlier notified varieties. Exactly. So the Government of India, the seed division of the Government Ministry of Agriculture, compiles the requirement of the breeder seed of different crops, and this process is a continuous process. It takes place 5 times in a year. for the Kharif season, for the Rabi season, for the agriculture and horticulture crops separately, and for the potato, they used to work separately. So they compile the requirement of the breeder seed and they forward it to the ICR through ADG seed and ADG horticulture. And the ADG horticulture and ADG seed forward into different PDPCs, the project director and project coordinators. Like in case of Maize, we are the project director, and we are responsible for the production of breeder seed as per the requirement of Government of India from the different indenters. So indenters are basically the seed producing agencies that are working in the country. So the work of the research and development and getting the getting the variety notified is being taken care of the one type of agencies, and the work of multiplication from breeder to foundation and foundation to certified is being done by the different type of agencies. So this type of system exists since the 1960s, the beginning of the 1960s, where the formal seed production system started. And in the government system, mostly this type of system exists in the government system. So once the production of the breeder seed is complete, after the indent is received, the production is being forwarded to the PDPCs, and PDPCs forwarded to different breeder seed production centers. Like us in the one of the this like RMR, Regional Maize Research and Seed Production Center of Begusarai, is one of the BSPC in case of maize. So we used to, at the ground level, the breeder seed production centers take up the breeder seed production as per the requirement of the different varieties, and finally the total production of the breeder seed of a variety, then of a crop, and then of all the crop that is being produced in a particular season is compiled by the different PDPCs, and the production is being forwarded to the ADG seed, and from ADG seed to the seed division. So previously there is a portal which was there in which all the activities is being taken up, was the SeedNet portal. But now the SATI, the Seed Authentication, Traceability and Holistic Inventory portal is there. Now all this activity of compilation of the breeder seed indent, forwarding it to the ICR, then after ICR to different PDPCs to BSPCs, and their monitoring, their processing, packing, tagging, and each and every activity is now being captured on the portal, because it is not possible to individually do the exercise or manually do the exercise. Previously, since 2008, there was a Government of India website, that is SeedNet, on which this exercise is being done by the different stakeholders that are involved in the system. But that was not a completely digital platform. Now, from 2023 onwards, the Government of India has launched another portal that I have already told, that is SATI, which is completely digital. So there is no need of pen and paper or any kind of physical inventory to be maintained. Everything should be kept on the work that has been done by the breeder, the seed producing agency, the seed testing agency is being recorded on the SATI portal, only. So the production that is being taken up by the different breeder seed production is being provided through SATI portal, forwarded to the seed division, and seed division, after getting the production in context to the indent, and as per the availability of seeds, suppose 100 quintal indent is being placed to the seed division of a particular variety, and they received the 80 quintal production only. So as per the requirement of the different agencies, the quantity of seed is being distributed, breeder seed is being distributed as per the availability. So the role of the seed division is the coordination between the ICR Institute and the indenters. The role of ICR and its institutes, including the SAUs and other institutes that is there in the system, is to produce the breeder seed of the varieties that is being developed by the particular institute. The role of the institute and this the PDPC is to get the variety produced that is being developed by the particular institute as per the requirement of the government of the different seed producing agencies. So after that, the role of the this research and development institute, SCUs, CAs are over, and the breeder seed is handed over to the seed producing agencies. So there are a number of because our country is quite diverse, and the seed production is being taken up by the different types of agencies. So in the beginning, when the formal system of seed production started in 1960s, we have the national level agency, National Seed Corporation, 1963. After that, there are 17 seeds production state seed corporations were established. And now, if we speak about of the current time, the different types of agencies are doing the seed production work. of this certified seed production, like National Seed Corporation, state seed corporations, even the ICR Institute is doing the foundation and certified seed production. State agriculture universities are also doing, and the Krishi Vigyan Kendras are also doing the seed production. Cooperatives, national level agencies like IFCO, CRIPCO, IFDC, one more organization, Bharatiya Beej Sehkari Samiti Limited, is doing the seed production work, and the private companies. Private companies, the small companies which don't have their own R&D, and they are wholly dependent. They are just doing the multiplication. They are basically taking the seed from the different institute that have developed the varieties, either through the Government of India seed division, through the national seed associations. Two national seed associations are there, which represent the stake of the private companies which have the requirement of the government bred varieties. So the intent of different private seed companies, you might be aware that there are MNCs working in the country, but if we talk about the small and large private seed companies in the country, there are approximately 10,000 companies that are working in the country. So not all the companies have their own R&D. So the small companies which are there, generally depends upon the seed that is being developed by the government agencies. So they do the multiplication and the marketing of the seed of the government bred varieties, and the private companies which have their own R&D do the entire work themselves. So the private companies either can generally produce the truthful label seeds, or they also produce the certified seeds, the small companies, and do the seed marketing and distribution. And finally, through this channel, the seed marketing distribution, the seed is available to the farmers. The foundation seed and the certified seed is being produced under the supervision of the seed certification agency. So seed certification agency is basically nothing. It is the agency that is responsible for the implementation of the different seed standards that is being developed by the Government of India. So the role of the seed certification agency is neither the seed producing agency or neither the seed producer. It is a third party which is responsible for the monitoring of the different parameters according to which the seed production is being taken up by either at the field level or at the seed processing and the testing. So all this, right from the seed production to the testing and packing, the supervision is being done by the seed certification agency. So basically, if we speak about of our country, there are 26 seed certification agencies in the country, in our country, and 3 of them are there in the Northeast. So most of the states have their own seed certification agency, and 3 states in Sikkim, Assam, and Arunachal have their own seed certification agency in Northeast, and they have their seed testing labs. There are approximately 175 seed notified seed testing labs in the country. The ICR institutes and the private companies might be having more labs, but they are not notified. So the testing of the certified and foundation seed takes place in the notified labs, which are generally there under the seed certification agency. After that, when the seed meets the field standards and the seed standards, it is finally tested and the certificate is being given to the concerned seed producing agency and is available for the marketing and distribution. And the seed is finally available to the farmers through the different marketing channels. So the seed system in India, the marketing and distribution. So if we speak about the marketing, so different types of agencies have their different channels of marketing. So marketing is basically the promotion or the outreach through which the new varieties comes to the system. So different methods are adopted for the marketing of the new varieties, because the farmer generally does not takes place the new variety itself. It has to be promoted, because changing the mindset of the farmer is the most difficult task. Though the farmer fairs, meetings, field level demonstration visits, government schemes, mini kits distribution, electronic and print media are the different types of marketing channels that is being adopted both by the private companies and the public companies. After that, the variety is popular or the seed distribution takes place. The government agencies usually follow the government organizations. The seed moves to the seed distributors, seed dealers, and ultimately to the farmers. Or they have a different other channel is the seed from the government organization to cooperatives or societies, and then it reaches to the farmers. Or sometimes the government agencies have their own sale counters, and finally through their own sale counters, the seed is being distributed to the farmers. The private companies follow the channel of the seed distributors, seed dealers, and ultimately the seed is reached to the farmers. So this is the overview of how the entire seed system, right from the development of the variety, the testing of the variety, and the production of the foundation of breeder seed, and involvement of different agencies, and how the finally the marketing and distribution takes place, and how finally the seed reaches to the farmers takes place in the country. This is the formal system that is being adopted by the different two types of agencies that are there, primarily there in the seed system. One is the R&D, research and development organizations. Other is the seed multiplication agency. Through this channels, all the notified varieties passes this channel and comes to the seed, and the final seed finally comes to the farmer. So this is the government sector system that works in the country for all the notified varieties for which the certified seed is being produced and provided to the farmers. So if we speak about the Indian seed network, I have already told that ICAR institutes are there, central agriculture universities are there, state agriculture universities, Krishi Vigyan Kendras, private seed companies are there, seed hubs, farmer participatory seed program. There are different schemes. One of the most popular schemes is the seed village scheme. National level organizations that are primarily dealing with the seed production, that is NSC was there previously. State Farm Seed Corporation was also there, but it was merged with the NSC. Now a new cooperative agency of the Ministry of Cooperatives is being launched by the Ministry of Cooperative, that is Bharatiya Bij Sehkari Samiti Limited. So these are the agencies at the national level exclusively working in the field of seed production. There are all the big states have their own state seed corporations, and the state seed corporation have the mandate to produce the seed of the particular as per the requirement of the particular states, and the national level agencies are there to produce the seed where the deficit is there at a state level, or for those states that don't have their own seed corporations. The public sector undertakings which are not directly there in the seed sector are NFL, IFDC, CRIBCO, NAFED, NDDB, HIL. They're also doing the seed production work, which is not their primary work, but they are there in the seed this seed system. So 26 seeds state seed certification agencies are there, and there are approximately 175 notified labs are there in the country. So if we speak about the breeder seed requirement, the government system, so data is called is little old, but the near about the total requirement of the breeder seed in the country is approximately the one that comes from the government of India, that is the indented breeder seed, and one that is directly sold through the this ICAR Institute or the breeder seed production centers, we say, because the breeder seed production centers are there with the ICAR Institute, SAUs, CAUs, and other institutes also, like NSC is also doing the breeder seed production. Maharashtra Seed Corporation is also doing even the BARC is also doing the breeder seed production. So the total requirement of the breeder seed is approximately 80 to 90,000 from the direct and indirect system also. And the total production is approximately 1 lakh to 1,10,000. Coming on to the availability and requirement of foundation seed, the total requirement of foundation seed is approximately 17 to 18 lakhs. The figures are old, but it is at present the requirement has increased. Approximately 17 to 18 lakhs of requirement of foundation seed is there for all those varieties which are produced through the formal system, and the availability that is there is approximately 24 to 25 lakhs of foundation seed is available. If we speak about the requirement of the certified and quality seed that is assessed by the different state agriculture department, that the total requirement of different certified and quality seed is approximately 500 to 510 lakhs lakh quintal of seed is required, and that much quantity is available with the different agencies in the country. And there is a big network that continuously works in coordination with the government of India, with the government, state governments, with the different government institute, with the ICAR Institute, Research Institute, and with these private companies that continuously works round the clock and in the entire year and repeatedly to provide the quality seeds to the farmers. Because there are different seasons, the requirement of different crops are there. So the government of India has the role to assess the requirement availability of seed, and total requirement generally increased by 5 to 6% every year. So the approximately 500 to 510 lakh quintals of seed is required in the country, and the same quantity is made available through the different organizations as I discussed earlier. So अब मेज की सीड की रिक्वायरमेंट की बात करें तो मेज का सिनेरियो सर ने जो the previous lecture was there, sir ने technically has elaborated each and everything, but the thing एक दो जो issues हैं that sir has not covered, I would like to cover, which are more practical, जो practicality जो issues हैं जो field level पे, उसको I would like to discuss. Rest most of the part is being covered by the sir. So as per the government statistics, the total requirement of maize seed in the country is approximately 17 to 18 lakh quintals. The exact figure is approximately 17 lakh quintals, but it varies year to year. And as per the information provided by the different states, the 94% quantity of the seed is being supplied by the private companies, and rest 6 to 7% is being provided by the government agencies as of now. So, ye issue, what is the issue that now this issue is not there in case of self-pollinated crop like wheat and paddy. This issue is reversed, and approximately 80 to 85% of wheat seed is that is being provided in to the farmers is being done by the government agencies and by the and rest by the private companies. So ये फिगर ऐसी क्यों है कि इसमें in maize की जो 94% ऑफ द जो सीड का सप्लाई है वो प्राइवेट कंपनीज ने कैप्चर कर रखी है और जो रेस्ट है वो गवर्नमेंट ऑफ एजेंसीज। और वन मोर इशू इज देयर कि जो सीड है वो बहुत कॉस्टली होता है इन कंपैरिजन टू द सेल्फ पॉलिनेटेड क्रॉप। वी स्पीक अबाउट अदर सीरियल क्रॉप दैट इज व्हीट एंड राइस। तो व्हीट एंड राइस की अगर हम कंपैरिजन करें तो the farmer used to get, if the market price of the wheat is approximately 25 rupees per kg, so farmer will going to get the seed in 50 rupees, maximum 50 rupees. Now some private companies have entered in the market, they are selling it 75 to 80 rupees per kg. So approximately the seed is costing 3 times more than the grain price. But in case of, if we speak about the maize के केस में जो बड़ा इशू तो नहीं कह सकते, मगर जो एक हमें जो कंपैरेटिव स्टेटमेंट जब हम फील्ड में जाते हैं, वी यूज्ड टू फाइंड आउट फ्रॉम द फार्मर्स की जो कॉस्ट है मेज ग्रेन की वो तो 20 to 25 रुपीस पर केजी है, लेकिन जो इन सम केसेस दे आर गेटिंग द सीड्स 800 to 900 रुपीस पर केजी पर केजी। और गवर्नमेंट का एग्जिस्टेंस ना के बराबर है, गवर्नमेंट सीड का एग्जिस्टेंस इज अप्रोक्सीमेटली नेग्लिजिबल। सो व्हाट आर द फैक्टर्स दैट आर मेकिंग the seed costlier, and why the government seed agency is not there in this maize system? So basically the issue is that कि maize का seed production तो sir ने I have discussed it elaborately in the previous lecture. So basically the issue is there that the production of maize hybrid seed is complex. Wherever the hybrid seed production system is there, whether it is in jowar, bajra, maize, the government agencies refrain from doing the hybrid seed production because it is a more tedious task and in more complex. So rather than doing the seed production in hybrid, they generally sticks to the open pollinated varieties. And क्योंकि maize में, because the seed production in maize is difficult if we compare it with the wheat. The seed production in wheat is quite simple. We just take the seed from the whatever seed producing agency that are willing to take the seed production. They provide the breeder seed. We do the multiplication, and again it provided to that particular agency as a foundation or as a certified seed. But in maize seed production, there are different factors that make it costlier. First, and the foremost, जैसे जैसे हम seed production के बारे में discuss करेंगे, तो that we will discuss how the cost of the maize seed is increased, and what are the reasons that making it the private the government agencies are not doing the seed production work. So if we speak about the government sector, the seed certification, they have to do the seed production through seed certification. So seed certification is the purpose of seed certification is to maintain the availability, make available the public through certification. High quality seed provide करना है certification agency का. But the private companies that are doing the seed production of the research varieties and the TL seed, they are not doing under the purview of the seed certification agency. If the government agency has to do the seed production and they have to do the certified seed production, then they have to do it through the seed certification agency. So seed certification agency का जो role है वो third party का है, that has to maintain the quality and to ensure that whatever the seed production is being taken up as per the minimum seed certification standards. So these are the different need of seed certification है, quality assurance है, genetic purity को maintain करने के लिए, consumer का protection है, so that whatever the seed production is being done, that the consumer should not be fraud, should उसके साथ कोई फ्रॉड नहीं होना चाहिए. So it should ensure that the proper system has been followed up to do the seed production. मार्केट एक्सेस तो सर्टिफाइड सीड कैन जो होता है जो उसमें बेटर मार्केट इट्स यूज्ड टू गेट बेटर मार्केट एक्सेस इन कंपैरिजन टू द मतलब unprocessed or non type of other type of seed, but it's not the case in each and every crop. Legal compliances है तो you all might be knowing that the our seed sector is governed basically by the Indian Seed Act 1996, and so whatever the legal compliances are there, the certification agency is bound to follow that and to ensure that they should be followed. So certified seed production, if it is there and it is provided to the different it is goes to the market, so it generally promote the research. The government में तो इसी basis पे research promote होती है कि the more and more certified seed is being given to the farmers, so more and more encouragement that the developers will get to develop the more varieties. Phases of seed certification है, receipt and scrutiny of application है, verification of seed source है, field inspection है, conformity to field standard, supervision of post harvest है, seed sampling analysis है, individual genetic purity and grant of seed certificate. So if we have to do the seed production in a maize or in any other crop that is to be certified, the variety should be notified and it has to be done under the purview of the seed certification agency, and they have to follow these steps to ensure that a certified seed is produced. There are two types of standard that is being observed in case of the certified seed production, whether it is foundation seed or certified seed, both types of standards are there. Field standards and seed standards. The field standards are observed at the field level, and the seed standards are observed after the harvesting is being done, so that the proper processing, testing, purity analysis should be done in accordance with the seed standards, and the seed that is being provided to the farmer should ensure that it sticks to the seed standards. सीड प्रोडक्शन मैं तो जेनरिक कवर करूंगा, सबने तो इलैबोरेट कवर कर ही लिया है. तो जो लैंड है शुड बी वॉलंटियर क्रॉप्स उससे होना चाहिए कि वॉलंटियर प्लांट्स नहीं होने चाहिए. प्रीवियस क्रॉप शुड नॉट बी ऑफ द सेम वैरायटीज, और मतलब whatever the land you are choosing should be free from volunteer plants or should be ensured that the same variety should not be there. फील्ड स्टैंडर्ड्स है तो इन केस ऑफ सर्टिफाइड सीड जो होता है वो F1 सीड होता है, जो हाइब्रिड सीड होता है. मेज में सर्टिफाइड सीड होता है वो हाइब्रिड सीड होता है जो फाइनली डिस्ट्रीब्यूट किया जाता है फार्मर्स को. तो उसके इन दैट केस 200 मीटर आइसोलेशन हैज टू बी मेंटेन. इफ द स्पेशल आइसोलेशन इज नॉट पॉसिबल, वी कैन फॉलो द टाइम आइसोलेशन. सो अब ये आइसोलेशन का जो फैक्टर है, ये जो 200 है या 400 केस इन केस ऑफ समटाइम्स, और अगर हम इसमें इनब्रेड जो अपनी नॉर्मल क्रॉप्स है उसके केस में कंपेयर करें व्हीट एंड राइस के केस में, if we compare to wheat and rice, it is only 3 to 5 I will be wrapping up in 5 to 10 minutes. So the isolation जो एक फैक्टर है मेज के केस में, that is making it difficult in comparison to the so more and more the maize का जो seed production है, it's more complex. Even the seed production is complex and seed processing is complex in case of open pollinated varieties. That is making the seed costlier, or it is making the seed costlier, and it is the reason that government agencies are not doing the seed production, because they don't want to involve in the complex system, because they have to follow the certification process. और अगर वो complexity इतनी अगर hybrid seed production में ले लेंगी तो they are not able to take up do the seed production. So that's why the government agencies का share जो है हमारे उस केस में, बेच के केस में, वो negligible होता जा रहा है. तो प्राइवेट कंपनीज डोमिनेट कर रही हैं, और जो इसका जो डिफरेंट चीजें हैं, जो डिफरेंट स्टेप्स है इसके प्रोडक्शन में, दैट मेक्स द सीड प्रोडक्शन कॉस्टली. फर्स्ट आइसोलेशन डिस्टेंस. अगर हम सेल्फ पॉलिनेटेड क्रॉप्स के बारे में बात करें तो उतना हमें नहीं चाहिए होता, बहुत इजीली हम कर सकते हैं. इवन इफ दे डू नॉट मेंटेन द आइसोलेशन डिस्टेंस, सो जो साइड की जो बाउंड्री के जो प्रोडक्शन होता है उसको आप पहले काट लें, और जो बीच में जो सेंटर पोर्शन में दैट इज लेफ्ट आउट, वी कैन टेक इट अप एज अ सीड इन केस इफ वी स्पीक अबाउट द wheat. So isolation distance I have discussed. Sowing, by sowing we have to do the sowing of 2 parents. If we compare about with the self pollinated crops, तो उसमें हमें सिंगल पेरेंट सो करना पड़ता है, इसमें complexity बढ़ जाती है. So 2 parents की sowing होगी, लेबर कॉस्ट बढ़ेगी, आपका टाइम कंज्यूम होगा, आपको मॉनिटरिंग वो होगी, मुश्किल होगी. So these are the factors at the field level that makes the things more complex in case of maize. So, and if the system is more complex, the system is more labor intensive, and you have to put more supervision, then the different steps, the complexity increase, and the cost of the production increases. So these are the factors that makes the hybrid seed production of maize more costlier in comparison to the other self-pollinated crops. इसमें मेल एंड फीमेल पेरेंट्स की सोइंग डिफरेंट डेट्स पे करनी है, डिफरेंट टाइम्स आपको प्रॉपर मेंटेन करके चलना है, रेशो मेंटेन करके चलना है। तो जितने भी आप जो ये प्रोसेस करेंगे उसमें लेबर इन्वॉल्व होगी और जितनी भी लेबर इन्वॉल्व होगी कॉस्ट बढ़ेगी इसमें। तो बॉर्डर रोज की सोइंग आपने सबने पहले बताई थी, तो बॉर्डर रोज की सोइंग करनी होती है। इसी की तरह से ये सोइंग फिर एक जो फ्लावरिंग है, फ्लावरिंग में जब हाइब्रिड सीड प्रोडक्शन पता आपको करना है कि जो फीमेल की डिटेसलिंग करानी है, तो डिटेसलिंग का जो प्रोसेस है उसमें भी आपको लेबर चाहिए और आपको पता है टेक्निकल का सुपरविजन चाहिए। तो मोर एंड मोर द सुपरविजन इज देयर, द टेक्निकल कॉम्प्लेक्सिटी इज देयर, द मोर द कॉस्ट इज देयर, द मोर द प्राइस विल इंक्रीज। So finally, the seed that is being produced as a female parent after crossing is called hybrid seed, that you all know. So इसमें stages of inspection is more. The more we have to do the inspection at four stages. In case of self-pollinated crop, we can do the inspection at 2 stages only. That will be suffice the purpose. But in case of maize, we have to do the inspection at 4 times. So that also increases the cost. Rogging hai, so rogging is basically done in all types of seed production crops. So that we have to do in the maize also. Detasseling, I have already told. Detasseling have to do. Seed standards. So the seed standard that is prescribed by the government of India in case of hybrid seed, the 98% purity is there and approximately 90%. So these seed standards should be followed up by whatever the seed, whether it is TL seed, whether it is certified seed going into the market. Ultimately, each and every agency has to follow, whether they have taken the seed production of the through certification or whether they have taken up the research variety seed production, that is TL seed. So I am again repeating that in production of seed, the certification is voluntary, but the labeling is compulsory, and the labeling has to be done as per the standard prescribed by the government of India. In case of hybrids, it is 98% pure seed and 90% germination should be there. Moisture 12% in case of vapor, जिसमें vapor आ सकती है, आपको जैसे कि 12% होना चाहिए, vapor proof containers में 8% होना चाहिए. Other crop seeds का minimum है. Whatever the things that improves the quality of the seed, their standards are prescribed as minimum, and whatever the things that deteriorates the quality of seed, their standards are prescribed as maximum. हार्वेस्टिंग है, हार्वेस्टिंग आफ्टर हार्वेस्टिंग, तो जनरली द हार्वेस्टिंग में कॉब्स की हार्वेस्टिंग होती है, फिर उसका मॉइस्चर जो एट द टाइम ऑफ हार्वेस्टिंग 20 to 35% होता है, तो उसका मॉइस्चर को रिड्यूस करके उसको लेके आना एक सेफ स्टोरेज पे, उसमें भी कॉस्टिंग आपकी लगती है। तो उसमें भी आपकी जो कॉस्टिंग लगेगी, आप आर्टिफिशियल, प्राइवेट कंपनीज आर्टिफिशियल ड्रायर यूज करती हैं, अगर जिसमें गवर्नमेंट जहां प्रोडक्शन कम हो रहा है वो आपको उसमें सन ड्राइंग के लिए भी जहां इतनी रिक्वायरमेंट नहीं है या छोटा प्रोडक्शन, सन ड्राइंग कैन आल्सो बी यूज्ड, बट टू रिड्यूस द कॉस्ट इज देयर एंड द क्वांटिटी आल्सो रिड्यूस। आफ्टर दैट क्रॉप की सॉर्टिंग है, डिस्किंग है और उसकी शेलिंग है, एंड आफ्टर शेलिंग द प्रोसेसिंग टेक्स प्लेस। प्रोसेसिंग के बाद जो अल्टीमेटली जो हमारी प्रोसेसिंग होती है, पैकिंग और ट्रीटमेंट होता है, पैकिंग होता है, स्टोरेज होता है, then finally seed is available to us. So if we basically start with 100 quintal of harvested crops, ultimately we are going to get 45 to 50 quintals of processed seed. So in case of if we compare it with any other crop like wheat, if we going to get raw seed 100 quintals, we are going to get 87 to 90% of the processed seed. So उसमें quantity भी reduce होगी. So these are the steps: the production, processing, drying, जो इसमें shelling है, फिर moisture का reduction है, जो quantity reduce होती है, that ultimately makes the seed production costlier. तो ये सब चीजें हमारी, these types of things are not there in self-pollinated crops. So these are the reason that increases the cost of the maize seed and the complexity increases, because of which the government agencies are refraining from doing the seed production. But as we are the institute that has the responsibility to develop the varieties, we have the varieties, but the indents of the different varieties that we have is not coming from from different seed producing agencies, because of which the market is being captured by the private companies. तो ये issues हैं जिसकी वजह से हमारा जो गवर्नमेंट का सेक्टर है, जो कंसर्न इश्यूज हैं, इसकी वजह से हमारा गवर्नमेंट का सेक्टर इतना प्रोलिफरेट नहीं कर रहा है, प्राइवेट का वो है। तो एक मैंने इंसाइट दिया आपको किस तरीके से हाउ द इंडियन सीड सेक्टर वर्क्स एंड जो मेज में क्या क्या इश्यूज हैं, क्या क्या जिसकी वजह से हमारे जो है जो गवर्नमेंट एजेंसीज जो हमारी हैं, वेदर इट स्टेट सीड कॉर्पोरेशन, जो प्राइवेट कंपनीज भी जो छोटे लेवल पे दैट डोंट हैव देयर ओन R&D एंड द नेशनल लेवल एजेंसी शुड कम फॉरवर्ड. We are there to support them, so come forward and to give the more and more Indians, so that the government का जो हमारा इतना डेवलपमेंट हम कर रहे हैं उसका कोई फायदा नहीं अगर अल्टीमेटली सीड जो किसानों को नहीं पहुंचता है. तो कुछ चैलेंजेस हैं टू डेवलप हाई यील्डिंग वैरायटी. ये तो कंटिन्यूअस प्रोसेस है, चलता रहता है कि मोर एंड मोर वैरायटी शुड बी देयर. डिजीज रेजिस्टेंस है, क्लाइमेट रेजिलियंस वैरायटीज होनी चाहिए. तो इट इज अ कंटिन्यूअस प्रोसेस फॉर प्रोवाइड क्वालिटी सीड टू द फार्मर्स एट अफोर्डेबल प्राइस. तो वेज में थोड़ा सा इशू रहता है कि जो द सीड प्राइस इज क्वाइट हाई, वो रीजन भी है उसका, और थोड़ा सा उसमें भाई किसान को अल्टीमेटली देखता है कि भाई रीजन उसको जो वो है, जैसे बिहार में हम बात करते हैं तो दे हैव द इशू दैट इंक्रीज ऑफ शेयर ऑफ द गवर्नमेंट सेक्टर बेड वैराइटीज इन सीड अवेलेबिलिटी. तो रिसर्च हो रहा है, लेकिन द शेयर ऑफ द सीड अवेलेबिलिटी जो हो रही है इन केस स्पेसिफिकली इन केस ऑफ मेज इज नॉट इंक्रीजिंग, व्हिच इज अ बिग इश्यूज. बट वी आर डूइंग आवर पार्ट. As I've already told you in the beginning, that this in case of government sector, this is entire scenario is divided into two types of institutes. The one is R&D, another is the seed producing agencies. So R&D part is being done by us, but the seed production part is not being taken up by those agencies. So यही challenges है इसकी और यही opportunities है. If we focus on this, so definitely we can contribute more. So with these words, I would like to thanks all of you. And if there is any question, you can ask. Thank you very much.
किसी का सवाल है तो पूछ सकते हैं सर से. Anyone?
नहीं रोमन, आई डोंट थिंक देयर इज एनी.
Okay,
जस्ट अ
sir.
रिक्वेस्ट. डॉक्टर विकास चंद्र कह रहे हैं कि हमें पीपीटीज ऑफ ईच सेशन, वो तो चलो वीडियो
PPT, sir, we are trying to collect some. We can get some. They would not like to share their PPT, sir. But the thing is,
वो डॉक्टर साहब हम कैसा करेंगे जब ये जो देगा कुलीग, एंड में हम वही 21 डेज वाला ग्रुप है ना व्हाट्सएप वाला.
Yes, sir. Yes, sir.
एंड में ट्रेनिंग खत्म होने के बाद उसमें डाल देंगे
Yes, sir.
यस.
Yes, sir. But other thing, what they can do is they can revisit the videos that we have uploaded in our
video
YouTube channel.
best. That is best. Yes.
So they can revisit anytime. So we have live telecast all the lectures in our YouTube page, so you can revisit anytime. So if there is no further questions, sir, I would like to thank Chandramohan sir for your excellent presentation, sir. And you have given an overview of the whole seed system in India. And thank you for your nice presentation. Thank you so much, sir. Sunil sir, अगर किसी का comment.
Yes. I had just one query.
Dr. Sunil, please.
Yes. So you have told about the some private company क्या कर रहे हैं कि अभी research hybrids ला रहे हैं. Okay. So rather than the private companies, the state seed corporations, they are also working on the development of maize hybrids. And suppose their hybrids it were not at par in the university trials, then these state seed corporations, वो उनका जो hybrid है वो research hybrids करके license मांग रहे. So can we give them the license?
आप सर कहाँ से हैं? उससे
महाराष्ट्र से.
डिपार्टमेंट से हैं?
नहीं महाराष्ट्र से.
नहीं डॉक्टर सुनील इज फ्रॉम आवर इक्रिप सेंटर इन
I am from इक्रिप सेंटर.
ऐसी आपकी जो MSSCL है that used to work in the some development उसमें, तो उनका तो मैंडेट ही है कि नोटिफाइड कराना ही कराना है. दे कैन नॉट गो फॉर द रिसर्च वो जो आपके रिसर्च वैराइटीज को करके आप प्रमोट नहीं कर सकते, क्योंकि दे आर द पार्ट ऑफ द गवर्नमेंट सेक्टर. अगर आप स्पेसिफिकली स्पीकिंग अबाउट द महाराष्ट्र सीड कॉर्पोरेशन ना, दे आर देयर इन द R&D. तो दे कैन नॉट, वो रिसर्च जो प्राइवेट कंपनीज कर रही है रिसर्च हाइब्रिड करके कर ले रही है, वो दैट दे कैन नॉट डू सच काइंड ऑफ थिंग.
Okay.
उसमें डॉक्टर सुनील लाइसेंसिंग कैन ओनली बी गिवन इन केस ऑफ नोटिफाइड पेरेंटल हाइब्रिड्स.
नहीं sir.
They can take up the seed production, TL seed production of the notified variety.
Notified, that's what I said. Notified only. The basic requirement is notification of the hybrid. फिर कोई भी class.
नहीं सर माय क्वेश्चन, मेरा क्वेश्चन ये है कि जैसे कि अभी प्राइवेट कंपनी भी क्या कर रही है कि उनका जो रिसर्च हाइब्रिड है, तो वो रिसर्च हाइब्रिड को वो सेल करने के लिए गवर्नमेंट ऑफ महाराष्ट्र को परमिशन मांगती है.
नहीं डॉक्टर साहब, मैं वही कह रहा हूँ, अगर वो रिसर्च हाइब्रिड भी है तो विच एजेंसी हैज आइडेंटिफाइड इट एक्सेप्ट दैट प्राइवेट कंपनी ना. तो आपका बेसिक जो सीड एक्ट का रूल ही है कि इन ऑर्डर टू बी कमर्शलाइज्ड इट हैज टू बी नोटिफाइड अंडर द इंडियन सीड एक्ट, अदरवाइज कमर्शलाइजेशन हो ही नहीं सकता. दैट्स अ बेसिक रूल.
नोटिफिकेशन के बिना वो जैसे सर का जो
हाँ सर वही है जो सर, विदाउट नोटिफिकेशन दे आर सेलिंग द हाइब्रिड्स.
प्राइवेट कंपनी जो
maybe Dr. Chandra Mohan can throw some light, otherwise notification I believe is the basic requirement, है ना.
मुझे जो समझ में आया ना सर, ये कह रहे हैं कि जैसे जो प्राइवेट कंपनीज है वो तो रिसर्च वैरायटीज का टीएल सीड बेच रहे हैं. अब गवर्नमेंट की जो वैरायटीज है, महाराष्ट्र सीड कॉर्पोरेशन, वो भी चाह रही है कि जो वैरायटी वो डेवलप कर रही है उसको बिना नोटिफिकेशन कराए रिसर्च के थ्रू बेच दें.
हाँ यही मेरा क्वेश्चन है.
सर एक्चुअली क्या है कि गवर्नमेंट एजेंसीज का मैंडेट है कि सिस्टम को फॉलो करवा के काम करना. तो अगर सिस्टम को बाईपास करने लगेंगे तो फिर तो कोई सिस्टम को फॉलो करेगा ही नहीं ना. ओके, तो ये मैंडेट है कि वो अभी प्राइवेट कंपनीज का भाई प्रोडक्ट बिकता है, आपका प्रोडक्ट बिक रहा है तो आपको
नहीं, मैंने उनको अपोज किया है, उनको मैंने नहीं दिया है. सो दैट्स व्हाई आई एम आस्किंग टू यू दिस क्वेश्चन.
वो दे हैव टू फॉलो द सिस्टम.
यस, ओके.
क्यों वो नोटिफिकेशन क्यों नहीं हो रहा है कि वो वैरायटी सुपीरियर होगी ही नहीं उनकी, अगर होगी तो जरूर होता उनका.
यस, ओके.
तो ऐसी वैरायटी तो हम पे क्राइटेरिया क्या है नोटिफिकेशन का, जो बेसिक क्राइटेरिया देखिए
वो क्राइटेरिया से नहीं हो रही है, इसलिए तो वो इस तरह से रहा है.
गेम से के लिए थोड़ी ना करना है उनको भाई, उनका अल्टीमेट गेम वो है, उनको प्रोवाइड करना है कि सीड प्रोवाइड होना चाहिए. अगर वो वैरायटी परफॉर्म करेगी नहीं तो क्या करना है उसको करना है उसको.
अगर और कोई क्वेश्चन, अगर किसी का क्वेश्चन नहीं है तो सर थैंक यू सो मच वंस अगेन सर. We will wrap up this session. We'll see you tomorrow again. Thank you so much.
Thank you everyone.
Thank you.